Okay, so after glycolysis, we have a step in between glycolysis and the Krebs cycle. And I think we talked a little bit about this the other day. That's called the preparatory step. And I want you to notice something about pyruvate. Pyruvate's got 3 carbons. What just happened to one of those carbons right here? Just lost it. Because what's happening to the pyruvate in this step? We're making NADH. Yeah, which means NAD plus got reduced to make NADH, which means what had to be happening to pyruvate? Oxidation. So pyruvate is getting oxidized, which means what just happened to some of the energy that was in the pyruvate? It got taken out and put where? That's what that's all about, NADH, storing energy that used to be in the food. So take some of the energy out of the food, some out of the pyruvate, put it in here. What's left of the pyruvate after this prep step started out with 3 carbons. What's left is a 2-carbon group called an acetyl group. That's 2 carbons. That combines with this substance called coenzyme A to make a chemical called acetyl-CoA. These 2 together. This actually enters the Krebs cycle. Very quickly, acetyl-CoA reacts with a substance called oxaloacetate to make a compound called citrate. That's so important that you know the names here. What you need to understand is that citrate goes through a series of steps where right here, what's happening to the citrate? Oxidation. Carbons broken off, and NADH is made. So NAD plus became NADH. NAD plus got reduced, therefore this stuff had to be being what? Oxidized. Keep going down, here's another oxidation step. Keep going down, here's another oxidation step. Because this FADH stuff is very similar. FAD plus some electrons plus some hydrogens give us this FADH2. So think of this pretty much just like the NADH. It's about storing what? 3 things. Electrons, hydrogens, and energy. And then we got one more oxidation step right here. But notice at the end of the Krebs cycle, what gets regenerated at the end? This oxaloacetate, and then it's one of the initial reactants to start back over, right? So it seems kind of wasteful that we're making a little bit of NADH, we even make a little bit of ATP down here, but not much. But the point being is it regenerates the beginning parts too. So we start off with this oxaloacetate, we end back up with it here at the end. That's why it's called a cycle. Starts over and over again. But the key things about Krebs cycle that you should know. Let me back up a slide. Try to back up a slide again. Think of Krebs cycle as the oxidation of pyruvate. And remember that oxidation is taking away, what, 3 things?
Electrons, hydrogen.
Yep, electrons, hydrogen, more importantly, energy. Taking away energy from the food. Make sure you know that the Krebs cycle happens in the matrix of the mitochondria. And that during that process of the Krebs cycle, hydrogens and electrons are stripped away from the pyruvates, and they're transferred to these electron-carrying coenzymes, either NAD plus or FAD. Now I'm lumping together in this slide Krebs cycle and the preparatory step. I'm going to kind of lump those 2 together. Preparatory step and Krebs cycle. Oxidation of pyruvate is what's going on there. So per glucose that we start with, during the prep step and Krebs cycle put together, we make a total of 2 ATPs per glucose. 6 CO2s are released, which makes sense because how many carbons did we start with in glucose? 6. So there they are. 8 molecules of NADH and 2 molecules of that very similar compound, FADH2. Remember, these are storing energy. energy that used to be in the food. Now here's another important point. After this is all said and done, after Krebs cycle, where's most of the energy at now that used to be in the glucose? Yep, in these things. So it's the job of the last step in cell respiration, electron transport, to get the energy out of these two things, and use that energy to make what? ATP. So the last step is called electron transport chain. Sometimes it's got another name, and this is a name that you see used more and more these days. Sometimes electron transport is called oxidative, because it's oxidation, phosphorylation. That phosphorylation word, well, my board's off up there, keeps showing up. Key thing before we even start going over any of this. During electron transport, two main things are being oxidized. NADH and what? FADH2. Those are getting oxidized. We're trying to get the energy out of those. And what are we trying to phosphorylate? ADP in order to make ATP. So that name is very descriptive of what's happening. NADH is getting oxidized in order to phosphorylate ADP so that you can make some ATP. Some basics of electron transport. First of all, we already said earlier that it happens on the cristae of the mitochondria. Why are those cristae so convoluted and twisted? More surface area, so you can do this the way you need to. Another important point. Do some bacteria do aerobic respiration? Can bacteria do aerobic respiration? Yep. Do bacteria have mitochondria? No, they don't have any organelles, right? So where does this happen in a bacteria? Turns out this part, electron transport, has to have a membrane, because it has to have proteins that are embedded in the membrane. What's the only membrane that bacteria have? Your cell membrane. They don't have a nucleus, right? They have the plasma membrane, that's it. So realize that in bacteria, this step doesn't happen in the mitochondria because there's not one there. The things that make this happen are all located in the cell membrane in bacteria. But the process is pretty much the same, it's just the location is different. In you, this happens on the membranes of the innermost part of your mitochondria. In bacteria, it happens on the cell membrane. But the outcome's the same. I'm going to draw a picture up here. Kind of a funny looking picture. But it's a picture that you need to be able to replicate. People make fun of this picture, but it's descriptive of what you need to know. Let me get a blank screen so I can do this. Just do it this way. All right, sometimes we call this picture the frog picture. Because here we go. What do you think this is supposed to be a picture of? A clam, right? A giant clam? Yeah, really, what do you think it's a picture of? It's supposed to be a mitochondria. This is supposed to be the outer membrane of the mitochondria. What would this area in here be? The matrix, and these are supposed to be cristae. Now, embedded in the membrane of the cristae are proteins. You have membranes embedded in your phospholipid bilayer too, right? Your cell membrane. Same thing here. These proteins have a special name though. These are called cytochromes. And these cytochromes have several jobs. Before we get to what those jobs are, though, let's start here inside the matrix. What is there a lot of in the matrix? Now, not the cristae, but inside the matrix. From the last step, what's the main thing that got made during Krebs cycle? NADH, and also the FADH too. But here's some NADH. That NADH is loaded with what? Energy. In this step, we need to get the energy out. This very first cytochrome number 1, will take that NADH and oxidize it. Oxidize simply means take apart what? Take away what? Right, energy, electrons, and hydrogens. The main thing the cytochrome takes away is the electron and the energy. So that electron, we're going to draw it out right there. There's the electron. It took it. What's left of the NADH once its electron gets taken away? Hydrogen and? What did it start out as? What did the NADH start out as? NAD plus, right? So it's back to that. So this is ready to be reused again, this NAD plus is. But the main thing is now the electrons up here on the cytochromes. What's the name of this step that we're talking about? Electron transport chain, right? So these things are like an electron transport chain. They move the electrons. Think of these little cytochromes like wire. What happens when you hook up electrons to a wire? They move through it. So they start to jump across it. The electrons are moving across the electron transport chain. Now here's the thing. As they're moving, what kind of energy do they have? Things that are moving have what kind of energy? Kinetic energy. That energy is used to power these proteins to do their other function. These cytochromes are also carrier proteins. What have we talked about with carrier proteins before? What do they do? What process? Carrier proteins. Active transport, right? Which means they pump things out. Yes, the gradient. And what these things like to pump is they like to pump hydrogen ions, H pluses, protons. Remember, an H plus is a proton. And the way they do that is they take H pluses from inside the matrix in here, and they pump them into this space between the two membranes. So they pump H plus into this space. So H plus starts to build up out here. Lots of H plus builds up in this space between the two mitochondrial membranes. Anytime an ion builds up, what ions want to do, I mean, if we personalize them here, make them human-like, if they were thinking, what are they thinking? Where do they want to go? They want to move, right? They want to move from what to what? High to low. Well, they're getting pumped out of here, out of the matrix, which means this becomes low, right? And out here is high. So what do they want to do? Back in. Why can't they? This membrane is made out of what? What are all membranes in the body made out of? Speak up. No. Phospholipids, right? Which are non-polar. These are ions. Do phospholipids and ions like each other? No. So they can't go back through. But it turns out there's another way in. And that other way in, we're going to draw in down here. There's another protein embedded in this cristae. This protein's got a special name too. It's called ATP synthase. ATP synthase. As soon as you see that name, it ought to be pretty obvious what it does. First of all, it's an enzyme, ASE. It's an enzyme that does what? Synthesizes ATP. Here's what happens. This thing is not only an enzyme, it's also a channel protein. There's another word back from a long time back. What do channel proteins let happen through them? Right, they let larger charged particles travel through them, and what's that process called? Facilitated diffusion, right? High to low. And guess what this channel lets go through it? H plus, the protons. When it opens up, these things rush in. H pluses rush in from the outside part of the mitochondria into the inner part, from high to low. And they rush in really, really fast because there's a really, really big what? Difference, concentration gradient. Remember, the bigger the gradient, the faster cell transport happens. If they're moving really fast, what does that mean they have a lot of? Energy, kinetic energy. This enzyme uses that kinetic energy sort of like one of those paddle wheels that you put into like a river. What did we do with those a long time back? And what do we still do with those paddle wheels? You let water run over one of them. Water's got kinetic energy. It turns the wheel, which is hooked up to what? A turbine, a generator, and that makes electricity. This is the same kind of thing. As these H pluses run through there, this thing grabs that energy and uses that energy to take ADP plus what? Plus P plus that energy in order to make ATP. And we call that phosphorylation, right? This got phosphorylated. ADP got phosphorylated in order to make ATP. Does that sort of make sense? I'm going to run back through the whole picture in a minute. But let me backtrack to another PowerPoint.
So the main job of the cytochromes is
It has two jobs. To move the electrons, number one, and to pump the H pluses. Now, one thing that we haven't talked about that we need to is this. If we think back to that picture for a second, we know that one of the reactants of our cell respiration is oxygen, right? Do we need oxygen to do glycolysis? Nope. Did we need oxygen to do Krebs cycle? Nope. Have we talked about oxygen yet up here? Nope. Turns out we don't need oxygen until the very last step. Wish I had. Let's see if I've still got my picture up here. I think I erased it. Yeah, I erased my picture, but just a quick redraw. All right, here are these cytochromes. The electron eventually makes it over here to that last one. If it doesn't have any place to go, what's going to happen when the next electron comes hopping across? It's going to be like a traffic jam. And eventually it just keeps jamming up and jamming up and jamming up, and what happens? What happens in a traffic jam? Everything stops, which means you stop doing cell respiration, which means you very quickly run out of ATP, and you very quickly die. Here's the only thing we need oxygen for. That oxygen gas that we breathe, its job is to take that electron. Oxygen is said to be the last electron acceptor, because that's what oxygen's good at, is taking electrons, right? Oxygen is one of the most, what, in the periodic table? Electronegative. It likes to take electrons. That's why it's called oxidation, right? Oxidation, it takes electrons. And it takes those electrons and it combines them with what? Hydrogens. And if you take some oxygen and some electrons and some hydrogens, what do you end up with? Water. That's where the water in our cell respiration equation comes from. The key thing, if you don't have oxygen, these electrons don't have anywhere to go. And what happens to cell respiration? It stops. You need to know that the reason we breathe, the reason we need oxygen, is just for this. So that oxygen can be the final electron acceptor. It can take those electrons away, turn it into water, so that cell respiration can keep going. That's why we need oxygen. Now I'm going to go back to my other PowerPoint for a minute. and pull up a couple of things. This one step of electron transport makes 32 ATPs per glucose. How many ATPs had we made before this step? Well, we made 6, but we really only had netted how many? 4. So we're up now to 36 total ATPs per glucose. That's the general amount we usually get. Per glucose, we get 36 ATPs. When you eat a candy bar, how many glucose molecules are we talking about in that candy bar? Thousands, maybe millions. So you multiply that by 1,000 or 1,000,000, now you're getting some serious ATP. Enough to make you work. Now, I want to mention just a couple of other things before I turn this off. Back a few years ago, there was this diet drug that came out. And the scientific name of the diet drug was dinitrophenol. People started taking this medicine and they started losing a lot of weight. It was working. The scary part of these medicines is that some of them they design to do what they do. Those don't scare me that much. But a lot of times, where does the medicine come from? Somebody just happens to have tried some chemical on something and saw that it did this. For example, this Rogaine stuff. What did Rogaine start out as? You guys know what Rogaine is, right? It's supposed to make your hair grow. Okay, bald guys. What did it start out as? Started out as a blood pressure medicine. And they noticed that the guys taking the blood pressure medicine started growing hair. Well, maybe that too. But they didn't really know why it made them grow hair. They just saw that it made them grow hair, so they started giving it to people to make it grow hair. Same thing happened here. They figured out that these people who took this chemical, where they started using it, I don't know. I don't know the backstory on this. But they saw that if you gave people this chemical, they started losing weight. So they started giving it to people, not knowing what. Not knowing how it worked, not knowing what it did. Had no idea exactly what was going on. So people lost a lot of weight. This drug became pretty popular pretty fast because it worked. There didn't seem to be any side effects, at least short term. But then people started taking the medicine long term, like 4, 6, 8 months, a year. And people on this drug started dying, just falling out dead. So then what did the scientists have to go do? They had to figure out what it was doing. And here's what they found out. They found out that what it was doing was making this inner membrane permeable to what? H+. So instead of having to shoot back through this ATP synthase, it could just shoot back through anywhere it wanted to go, which meant it wasn't going through here, which meant what wasn't being made? ATP. It took a long time being on this drug for it to affect all your cells. So you were okay. But what was ended up happening is that a lot of your food energy was just being wasted and never being used to make ATP. So you just kept having to speed up respiration faster and faster and faster. And it got to the point where eventually the drug had affected enough ATPs, enough cells rather, enough mitochondria, that you couldn't make enough ATP to live. So you just fell out dead. It was like somebody was choking you. Literally, the drug was doing the same thing to you that choking would be doing to you. You're running out of ATP and dying. Kind of scary. And that's the scary thing about a lot of these new drugs. Even like antidepressants. If you watch those commercials for like Prozac or all those different ones on TV, they say, this is how we think this drug works. You don't really know. Personally, I don't really want to take something they don't know how it works. Because if they don't know how it works, they also don't know what it might do to you in the long term. That's the scary part. And that's the scary part about any new drug is that they're not really sure long term what it might do to you. All right, I'm going to stop there for a second. Yeah, you should know it just as an example.